In this module, I'm going to help you revise the topic on sexual reproduction and meiosis. Some organisms can reproduce asexually. Unless mutations occur, this generally produces clones of genetically identical individuals. However, this lack of genetic variation can be a disadvantage. Sexual reproduction involves the production of specialized sex cells called gametes and the fusion of their nuclei in a process called fertilization. Male gametes are smaller than female gametes and they are motile, which means they're able to move. There are many more male gametes produced. The female gametes are larger with more cytoplasm. Cells with the full number of chromosomes are described as diploid, abbreviated to 2N. If two gametes are going to fuse, then the chromosome number in the gametes must be halved. They must be haploid, or N, so that at fertilization, the diploid number of chromosomes is restored. So a special type of nuclear and cell division is needed, meiosis. The word meiosis means reduction. Meiosis involves two divisions, meiosis 1 and meiosis 2. Each division has 4 stages: prophase, metaphase, anaphase, and telophase. During interphase, before meiosis 1 begins, the DNA replicates. During prophase 1, the chromatin condenses and coils, and will take up stains, so that the chromosomes, each consisting of 2 chromatids, are visible. The nucleolus disappears, and the centrioles move to opposite poles of the cell. The nuclear envelope breaks up. So, the early part of prophase 1 is similar to prophase of mitosis. However, during the middle of prophase, the homologous chromosomes pair up in a process called synapsis to form bivalents. During late prophase, these bivalents exchange genetic material in a process called crossing over. Chromatids of the maternal chromosome cross over chromatids of the paternal chromosome, and the point where they actually cross is called the chiasma, plural chiasmata. Later, as the chromosomes separate, the chiasmata break, leaving swapped alleles. This increases genetic variation in the cells resulting from meiotic division. By the end of prophase 1, a spindle has formed. In metaphase 1, the bivalents, still with their chiasmata, attach via their centromeres to the spindle at its equator. In anaphase 1, the spindle threads contract, but the centromeres of the chromosomes do not split. Members of each homologous pair of chromosomes are pulled apart towards opposite poles of the cell. The centromeres go first, so they still assume the V shape. In telophase 1, the spindle breaks down. 2 new nuclear envelopes reform, cytokinesis occurs, and 2 cells with half the number of chromosomes are produced. Each chromosome still consists of 2 chromatids. In many plant cells, there is no cytokinesis at this stage, and they go straight into meiosis 2, whereas animal cells may have a short interphase, but no further replication of DNA before entering meiosis 2. Meiosis 2 is like mitosis, but starting with half the number of chromosomes. In prophase 2, the 2 nuclear envelopes disperse. The centrioles move to opposite poles of each cell, and the nucleoli disappear. A spindle forms in each cell, and note that this spindle is in a plane at right angles to the spindle of the original cell in meiosis 1. In metaphase 2, the chromosomes attach by their centromeres to the spindles at the equators. In anaphase 2, the spindle threads contract and pull the chromatids apart. The centromeres split first, and chromatids are pulled to opposite poles. In telophase 2, 4 new nuclear envelopes reform, and the nucleoli reform. The spindles break down, and the cells undergo cytokinesis. In animal cells, the cytoplasm cleaves, but in plant cells, an end plate is formed, and a wall forms on either side of it. The products of meiosis are 4 haploid cells, genetically different from each other and from the parent cell. Meiosis produces genetic variation in the following ways: crossing over at prophase 1, independent assortment of chromosomes at metaphase 1, and independent assortment of chromatids at metaphase 2. Sexual reproduction also produces genetic variation, as 2 haploid cells from 2 different individuals are fused during fertilization. This is usually a random process. We usually think of meiosis in conjunction with sexual reproduction to produce gametes. We can think about the life cycle of an animal, such as man, and indicate where mitosis and meiosis occur. However, things are not always this straightforward. Male honeybees, or drones, are produced from unfertilized eggs laid by the queen. This means that all cells in the drones are haploid. Drones therefore produce sperm by mitosis. Each queen is fertilized by a few drones. She stores the sperm and lays fertilized eggs that develop then into female worker bees. Genetic variation in the sperm arises because there is genetic variation in the drones. This variation is the result of eggs produced by meiosis in the queen. The life cycle of the queen bee and of the male bees is therefore different. Female worker bees, although diploid, do not undergo meiosis and do not produce eggs, as their reproductive organs are undeveloped. Ferns are terrestrial plants and have stems, leaves, often called fronds, and roots. The life cycle of ferns has 2 independent phases. The spore-bearing or sporophyte plant, this is the plant we call the fern, and the tiny independent gametophyte plant that produces male and female gametes. Spores are produced asexually by the sporophyte. The gametophyte can reproduce sexually. This diagram shows the life cycle of a fern plant. Meiosis is involved in spore production, and the resulting spores are haploid. Many of these are produced, and they are wind dispersed. So here, meiosis is involved in asexual reproduction. The spores germinate into the tiny gametophyte, which can produce haploid gametes by mitosis. When 2 gametes fuse, a diploid zygote is produced, which grows into a diploid sporophyte. So here, mitosis is involved in sexual reproduction.